Good morning, everybody. Good morning. My name is Jim Hood, annual meeting co-chair. I hope you've been enjoying our program of plenaries and special sessions. What? I hope you've been enjoying our scientific program and plenaries and the contributed session so far. This afternoon we'll be holding our only poster session of the conference in the ballroom 111 ABC. That's not this ballroom, it's the ballroom over there across the way. Please come and support our poster presenters who are eager to share their science with you. And if you're a poster presenter, please make sure you know which of the two poster sessions you're in. Odd poster numbers present in session 1 from 3:30 to 4:15. Even numbers in session 2 from 4:45 to 5:30. So please come and support our poster presenters. There we go. Also, please remember that the taxonomy fair is happening in the same location at the same time. If you're having a difficult specimen that you would like help with identifying, or you want to geek out on aquatic invertebrate taxonomy, please come and visit with one of our many experts. If your work is supported by NSF, or you're interested in seeking funding from NSF in the future, please come to today's Lunch and Learn with NSF Program Director for the Division of Environmental Biology, Dr. Robin Smith. This will be right here in the ballroom from 12:30 to 2 p.m. If you've been thinking about joining the SFS Non-Perennial Ecosystems chapter, there's a mixer for current and prospective chapter members today from 12:30 to 2 p.m. in room 201. That should be pretty good. A reminder that tonight is the social from 7 p.m. to 10:30. It will be located off-site at the Northwest Museum of Arts and Culture. You'll receive your drink tickets on site, so don't worry about forgetting them in your hotel room, but please bring your conference name badge. Also, we're asking attendees to find their own way to the event, and there are a number of options. It's about a 20-minute walk, it's about a $10 Uber, so that if you share with 4 people, it's basically nothing. For the return trip, we have a limited bus going back here for 3 trips at 9:30, 10, and 10:30. There are limited spots on this bus, and we want to save it for people with mobility issues, so just keep that in mind. All right, another reminder that the SFS Fellows will be meeting today over lunch from 2 to 2 p.m. in room 205. If you're a fellow, you'll know what that's all about. All right, so exciting news now. Yesterday at the membership business meeting, we issued a challenge, a fundraising challenge to match the generous $10,000 gift provided by the SFS Fellows to support our InSTARS, Emerge, HLA, and STREAM programs. And everybody really rose to the occasion. I'm really excited to report that we have met and exceeded our goal and have raised $10,295 in the last 20 hours. That's so awesome. So we really appreciate your support for the society. And I would say, why stop there? There are QR codes around the conference center, I think on the tables still. So if you haven't given yet, please, and you're able to, please take the time to provide to support these programs. They're really a wonderful thing that SFS is doing. All right, and then finally, I'd like to thank our sponsors and exhibitors. These entities make the meeting more affordable for all of you through their financial support. So let's give them a round of applause. And if you have time, please go out and visit them and see what they have to offer. All right, so on with our program now. Next, I'd like to welcome up A.J. Reisinger to talk about the Heinz Award. Thank you.
The Heinz Award for New Investigators is awarded to a freshwater scientist within 5 years of their terminal degree who was the senior author of an outstanding primary publication that appeared in print or online within the last 3 years. It is my pleasure to introduce the 2026 recipient of the Heinz Award, Dr. Audrey Geckner. Audrey is currently a postdoctoral researcher at the University of California, Santa Barbara. She received her dual bachelor's degree from the University of South Florida and her MS and PhD from the University of Florida, with a pretty good advisor. She received the Heinz Award for her 2024 paper, Subtropical Stormwater Ponds Are More Frequently Net Nitrogen Fixing Compared to Natural Ponds, published in Biogeochemistry. In this paper, Audrey presented the first study of net nitrogen gas flux in urban water bodies to estimate ecosystem scale net denitrification. Unlike prior studies from agricultural ponds and lakes, Audrey found that urban stormwater ponds are equally likely to be net nitrogen fixing or denitrifying, meaning that these engineered ecosystems are often a source of bioreactive nitrogen to the watershed rather than a sink. As you will see from her presentation, this paper has significant management implications. In Florida, stormwater ponds have been incorrectly presumed to remove 80% of the nitrogen inputs they receive from the developed landscapes they drain. This paper provides a biological mechanism for why these systems often don't work the way we expect them to. This work derived from her dissertation at the University of Florida, where she broadly focused on the biogeochemistry and microbial ecology of stormwater ponds, including carbon emissions, whole pond metabolism, and microbial community coalescence. Beyond her research, Audrey has been a fantastic mentor to undergraduate and graduate students during her time as a PhD student and postdoc, including an undergrad that contributed to her 2024 paper and continued in the field, and I think is in the audience today. Hi, Annabelle. Over the past few years, Audrey has moved from artificial ponds to working on terrestrial aquatic connections in Floridian and African wetlands. And her current postdoc work actually has taken her in a very different direction, where she is focusing on methane production in termite mounds in the Congo. Audrey was an excellent student and is an excellent collaborator, colleague, and friend. Also, just as an FYI, she's starting up a lab at Louisiana State University in the fall of 2027. So if you're interested in working with Audrey, I'm pretty sure she'd be happy to talk with you. Please join me in congratulating Dr. Audrey Geckner, our 2026 Heinz Award winner. And is Dr. Gessner in the audience? Can you come up and give the award as well and get a picture?
Hello. Hi, everyone. My name is Audrey, currently a postdoc at UC Santa Barbara. And today I'll be talking about nitrogen cycling in human-made ecosystems and how we've compared that to natural analogs. But first, I want to start by giving thanks to the Society for Freshwater Science for being an academic home for me since 2020, my first meeting being virtual. But my next meeting being the very big and amazing Joint Aquatic Sciences meeting in Grand Rapids, where I actually presented a preliminary version of this work. So it's an honor to be here today to share this with you again in this context. Also, a special thanks to the Heinz Award Subcommittee for recognizing this work that was a part of my dissertation at the University of Florida. And I want to quickly highlight my co-authors that were part of this work, Meredith and Ashley, who were incredible mentors to me during my program. And of course to A.J. Reisinger for being an extraordinary advisor. He continues to be a great mentor to me and has really shaped the scientist I am today in ways I cannot fully put into words. All right, so I'm going to start big scale here for just a moment. This recent map beautifully displays wetlands and lakes on Earth, and many of us study how these terrestrial kidneys enhance water quality through sediment, nutrient, and pollutant retention, or how they play a role in our global climate. But as our population has grown, the extent of our natural landscapes has declined. This map highlights the loss of wetland area since the 1700s, and just in the last 90 years, we've lost 35%. Meaning we have lost so much of the ecosystem service potential that these natural spaces have provided. It may not be surprising that the primary drivers of this loss include agriculture and urbanization. Focusing on urbanization, when we develop and pave the landscape, we need to construct artificial ecosystems to provide specific societal services similar to the natural ecosystems they are built to mimic, like the storage and conveyance of water and the retention of sediments, nutrients, and pollutants. So as natural spaces are in decline, human-made infrastructure is increasing in area, sometimes replacing natural wetlands in greater densities. As an example, here is Bradenton, Florida in 1984. This is close to where I grew up. And here is Bradenton now. The wetlands that human-made ecosystems replace provide rich benefits to society, nature, climate, and water quality. But do human-made ecosystems provide the same? In Florida, our stormwater rules require any new development to manage stormwater runoff through engineered systems like stormwater ponds, which are built primarily to prevent flooding and protect downstream water quality. These ponds must be designed to a particular size and volume depending on the size of the urban watershed. And like in that photo in Bradenton, stormwater ponds are heavily distributed throughout the entire state, with 76,000 plotted in blue here on the map on the left as of 2012, and that number is likely well over 100,000 today. And for a long time, we've been building these ecosystems with little knowledge of what they truly do in terms of water quality. So since the early 1980s, if you built a stormwater system, it was presumed that the discharge would not violate water quality standards. The performance standard was set at 80% reduction of suspended solids, and it was assumed that the same would be true for nitrogen and phosphorus removal. And a classic study by Harper and Baker went out and tested whether stormwater ponds met these requirements, and they used a mass balance approach to look at nitrogen and phosphorus removal efficiency across ponds of varying water retention times, shown here on the x-axis, and total phosphorus shown on the y-axis, removal efficiency. So when it came to phosphorus, they found that stormwater ponds, they found that stormwater ponds removed about 70% total phosphorus on average. And that's pretty close, so that's not bad. But total nitrogen, however, did not show the same result, removing on average 37% total nitrogen, regardless of how long that water was held in the system. And so they showed that the standard stormwater pond design was not removing that assumed 80%. So these stormwater ponds are designed by engineers to capture and treat runoff, but the design criteria assumes certain biogeochemical processes are happening inside, like denitrification, which is a nitrogen removal pathway. So, but they assume that without ever having measured it. So why aren't stormwater ponds efficient at removing nitrogen? We wanted to understand more about the nitrogen cycling processes that were occurring within these stormwater ponds that might help explain the mass balance findings. And since stormwater ponds are designed to mimic natural ecosystems, we wanted to understand more about how they measured up. To do this, we surveyed stormwater ponds in South Florida and natural undisturbed ponds in North Florida. And if you're wondering why they are so separated in space, it is really hard to find natural undisturbed ponds in Florida of the same size. Our natural ponds are further split between natural humic ponds, so rich in colored dissolved organic carbon, and natural clear water ponds. And we visited these sites during a dry season and a wet season. At each site, we went out and we collected water samples from 3 depths of the water column, depending on thermal profiles. Most of our ponds were mixed or weakly stratified. And I want to give a shout out to Annabelle Schreiber and Katie Schoenberger for the field support that they provided for this project while they were undergrads in the Reisinger lab. Okay, so with these water samples, we measured the saturation of N2 gas, and that's because N-fixation and denitrification are 2 dominant and opposing nitrogen cycling pathways that influence the balance of N2 gas. So nitrogen fixation is considered an introduction of nitrogen into an ecosystem where microbes convert N2 gas into tissue or bioavailable nitrogen. And whereas denitrification is considered a nitrogen removal from the system because it converts nitrate into N2 gas, which can then leave the system. And so we measure N2 gas and we compare it to how much should be there when in equilibrium, with that equilibrium value being 1. And so if it is 1, it's considered a balance between these 2 processes. But if we measure N2 and it is undersaturated and that ratio is less than 1, we consider that net nitrogen fixing. And if that saturation ratio is above 1, it's supersaturated, we consider that net denitrification going on. And this approach assumes to integrate processes at the ecosystem scale. Okay. Here's the data. We are looking at the frequency of N2 saturation ratios. And so if the data is below 1, it is net nitrogen fixing, and if the data is above 1, it is net denitrifying. And so in natural ponds, shown in green, a majority of the data is net denitrifying. And that number, 82%, is about what we've seen in temperate lakes. In stormwater ponds, however, half of the data is net denitrifying and half is net nitrogen fixing. So we are equally as likely to find a pond adding nitrogen into the system as we are to find one removing. And when we look at the saturation of N2 spatially in each pond type, the shades of blue here indicating water column depth, we see that the degree of denitrification increases with depth in the natural ponds as we move closer to the sediments, and that is exactly where we would expect more denitrification to be occurring. Whereas in stormwater ponds, we find net nitrogen fixation in both the surface and the near sediment area of the water column, and so this 50-50 proportion persists throughout the water column. And so with this data, we found that stormwater ponds are more frequently net nitrogen fixing compared to naturally occurring ponds. Using surface layer data, we can calculate a net air-water flux of N2, which gives us insight into the ultimate sink or source status of a pond. Again, you could find a stormwater pond removing as much nitrogen as another one could be removing it. But still, our net denitrification rates are very low compared to what has been recorded in one other study that has looked at this in residential ponds of China. And weak denitrification in Florida ponds has been seen in other cases in previous Reisinger lab members that use sediment core incubations. So in the left plot, under ambient conditions on the left, denitrification or the positive N2 flux is low and sometimes negative, so net nitrogen fixing, and there's low nitrate uptake. Whereas when we elevate the concentration of nitrate in these cores, that is when we start to see denitrification. So these systems need a sufficient amount of nitrate before they start denitrifying. Okay, so we know stormwater ponds receive plenty of nutrients and resources from the landscape, so why are they fixing nitrogen? We have a few theories. The first, let's take a look at the forms of nitrogen that stormwater ponds receive. A majority is organic, and so this study was done in the same area of Bradenton, and in our experience, we rarely find detectable or meaningful concentrations of inorganic nitrogen, despite there being high algal biomass. So we think that any form of inorganic nitrogen is rapidly taken up into plant or algal biomass. If you recall, stormwater ponds are very efficient at trapping phosphorus in sediments. Nitrogen fixation is typically occurring under low nitrogen to phosphorus ratios. So this large pool of phosphorus in sediments is likely driving down that N to P ratio. And finally, it's pretty common to find an algal bloom in a stormwater pond. And a survey of about 70 blooms in North Carolina found that a majority of these blooms were dominated by nitrogen-fixing cyanobacteria. So all of the prime conditions are there: high algal biomass of nitrogen fixers, competition for any incoming inorganic nitrogen, and a high pool of phosphorus in sediments that drive down that NP ratio. So these are highly productive ecosystems, and we have work coming out soon showing that they can be metabolically intense as well. All right, so what has changed in the policy and regulation surrounding stormwater treatment? In the past, we assumed all ponds removed 80% of incoming suspended solids, nitrogen and phosphorus, with only 37% of that nitrogen actually being removed on average. And now we know it's likely because microbial activities are fixing nitrogen and introducing it. But now our work ended up playing a part in a long discussion about stormwater management. And so a year after our work was published, Senate Bill 7040 was signed and implemented, which updated the state's stormwater rule. So for new stormwater projects, the general standard now requires nutrient reductions of 80% total phosphorus, so that's the same, but 55% total nitrogen from a proposed project and 45% from pre-existing projects if they are in an area of waters of concern. And what does this look like in practice? Instead of just building larger ponds, it means robust project-specific approaches to cleaning water. That means incorporating best management practices like vegetated swales, dry retention ponds, rain gardens, or emergent technologies, or sometimes a combination of these to create treatment trains. And so instead of creating a pond and just hoping for the best, this bill means conscious and intentional approaches to cleaning our water. Okay, so to summarize it all, artificial ecosystems are still ecosystems, and it's time that we start treating them that way. We found frequent net nitrogen fixation going on in these stormwater ponds, which is likely offsetting their ability to remove nitrogen, the thing that they were designed to do. We designed ponds by volume, not for biology, so meet the size specification and get the nutrient removal credits. But as we've seen, the biogeochemistry inside doesn't follow the engineering drawings. This work supported a broader shift in how Florida regulates stormwater from presumed compliance to requiring demonstrated performance under Senate Bill 7040. And looking ahead, human-made aquatic ecosystems are expanding globally, and so if we want these systems to actually deliver the ecosystem services that we're counting on, we need to start measuring what's inside of them. So these are ecosystems capable of providing similar functions to their natural counterparts if we let them. So thank you so much for having me here. I really appreciate it.
All right, so today we're going to have two plenary speakers again, Dr. Angus McIntosh and then Dr. Faith Kearns. And what we'll do is I'll introduce Dr. McIntosh. Then Camille will introduce Dr. Kearns after Dr. McIntosh speaks, and then we'll take questions at the end of both of their talks. So I'm pleased to introduce Dr. Angus McIntosh. He's a freshwater ecologist from the Department of Biological Sciences at the University of Canterbury, Christchurch, New Zealand. Dr. McIntosh is the co-leader of the Freshwater Ecology Research Group and also an associate dean in the Faculty of Science. His research combines investigations of food webs with spatial analysis and experimental approaches to reveal how environmental change affects freshwater ecosystems. He has published well over 100 peer-reviewed publications in journals such as Nature Geoscience, Ecology Letters, Global Change Ecology, as well as Freshwater Science and the Journal of the North American Benthological Society. Dr. McIntosh has won several teaching awards, including the National Award for University of Teaching in 2007. In 2023, he was awarded the New Zealand Freshwater Sciences Medal for his sustained and substantial contribution to advancing freshwater science in New Zealand and internationally. Dr. McIntosh has been a member of SFS since 1991, and he's also an amazing photographer, as you'll see. So please help me welcome Dr. Angus McIntosh.
Tēnā koutou katoa, kei te mana whenua o tēnā rohi, tēnā koutou. Nō Rakiora hau hi Pākehā ahau, ko Angus McIntosh tāko ingoa. Nō reira, tēnā koutou katoa. Greetings everyone. I've just greeted you in Te Reo Māori. I've acknowledged the traditional people of this land. I said that I came from Rakiora, which is an island down the south of Aotearoa, New Zealand. I said that I'm Pākehā, so a New Zealand European, so not Māori. And I come from Te Whare Wānanga o Wataha, so the University of Canterbury. It's wonderful to be here, and thanks very much for the invitation. Resilience and recovery is something that we all understand and we can all embrace. And it's a concept that the public, governments, policymakers embrace too. And so it's one of those concepts which on the one hand is simple, but has lots of complexities underlying it. And I'm going to encourage you to embrace those complexities, all of the dimensions of resilience today, and I want to run through some of those. So that's essentially what I'm going to try and do. There have been a lot of review papers and examples of work which has really sought to try and explain those dimensions. I'm not going to go into those aspects too much, but I want to lay out a framework at the start here. And so on the one hand, resilience can be about resources, recruits, refuges, so the ability of organisms to be able to come back, recolonize, recover, and the ecological processes which underlie that. Those are very important. Secondly, as freshwater ecologists, as stream ecologists, we all know that rivers are really dynamic, the flow regimes are incredibly important, and so those aspects, particularly the heterogeneity of those systems, are really important, and that's been characterised as, river corridor absorbing capacity, the capacity of a river ecosystem to absorb change, to be able to recover and retain its functions. Finally, there's a dimension which really represents resilience as a self-perpetuating system. Sam Blake brought to us to the fore, the late Sam Blake, an Australian freshwater ecologist who left us recently. Sam said a major goal in restoration is to restore both the resistance and resilience so that the restored entity is self-perpetuating and does not require ongoing interventions to be sustainable. So those three kind of approaches and views of resilience really cover the spectrum. And the last one especially focuses on human efforts, what we might want. It invokes sustainability, and that's a very important idea. So I want to, you to think about and to embrace all of those dimensions. This is my colleague, John Harding, and this might sound a bit cheesy, but I'm going to go with it anyway. John, very often when we're teaching classes, John teaching a field course here, you've got typical things going on. Someone's adjusting their hair. So if you're adjusting your hair now, pay attention. And there's someone, who's going, is this guy for real? But very often, things that are important are hiding in plain sight. And to see the light, we just need to turn around and look at it. So I'm kind of hoping that some of that might apply today. So pay attention, and we'll see if I can get you to see the light. We're going to have a few pictures of cool stream bugs. This is one of them, Zelander psyche ingens. It's a shredding caddisfly, which is common in forested streams in New Zealand in the South Island. And we did a crazy experiment which indicates the first thing I want to talk about, which is that the fundamental mechanics of resilience involve population regulation. This was a crazy experiment. It's the closest I've come to giving, didn't intentionally try to do this, but one of my field assistants, Hamish Gregg here, when he was undergrad, of giving him hypothermia. In fact, I think we both came close during this experiment. But what we did is we moved caddisfly larvae from 3 removal streams and added them to 3 addition streams and compared that to 3 control streams. And we did that in 200-metre reaches. And that was kind of a crazy idea, but it was possible because the caddisflies accumulate in leaf packs. So you can go up and pick up a leaf pack and grab a whole lot out, and they're big, like think cigar size. Haven't actually tried to smoke them, but they have cases about that big, so really huge. What we did is we did the manipulation just prior to pupation. And the graph there shows that we, in the removal streams, we halved populations. In the addition streams, we doubled populations. And the control streams, they stayed about the same. And those manipulations stayed in place 1 week, the white bars, and 3 weeks, the black bars, after the manipulation. And so then they pupated, and then they produced eggs. We were able to sample the adults and sample the eggs because the adults land on emergent rocks, and they have these big bulbous egg masses which you can go along and count. And then the early instars are really difficult to count, so Hamish did most of the really intensive lab work here of trying to pick them out of leaf packs when they're tiny first or second instars. Then we saw the results. And the results were pretty much like this, that there was absolutely no effect of any of the treatments. I think that happens a lot. I was kind of disappointed, but in the end, actually not surprised. If you have a look at the white dots there, those are the number of larvae that we estimated were put into the streams based on the number of egg masses we counted. No treatment effect. And then a whole lot of larvae were lost, and then the populations show this trajectory as we go from first instars through to the last instar there. And some streams gained larvae, some streams stayed about the same, and some streams lost larvae. And so this is pretty much representative of the stochastic dispersal processes. The number of larvae or eggs delivered to the stream had little influence on the eventual populations. And there was a really important process going on here that was density dependent, that the number of late instar larvae that survived was related to the number of early instars there, the survivorship was related to the number of early instars. And when you had lots, relatively few survived, and when you didn't have very many survived. And this was driven by resource supply, the abundance of high-quality leaf resources. Who thought that detritus would be in short supply in streams like this? And so this is indicative of density-dependent population regulation driven by resource abundance. And, as stream ecologists, we focus on the dynamicism, the disturbance, and lots of those stochastic processes that go on in streams. But very often I think we forget about the absolute fundamentals which we learn in first-year ecology, which are related to population regulation and those fundamental processes. And actually, interesting, I think we're coming back to those as we look to try and forecast in a non-stationary environment where vital rates and those fundamental processes are going to be really important. So the first thing I want you to remember is density dependence. The second thing I want to talk about is negative resilience. Sam Lake popularised this. Well, before he died, actually, I sent him an email and he said, I don't think that was a very good paper, didn't get very much attention, no one really believed me. But I'm a believer, and I want to explain it to you now. So Sam Lake said, resistance and resilience may be viewed as being positive in that they serve to maintain the restored state. He was talking about restoration. But he also said the resistance and resilience of the degraded state may be viewed as being negative in that it will serve to limit the effects of restoration interventions. And those of you who have been involved in restoration will know that we have a pretty high failure rate in stream ecosystems, and very often there's lots of processes which are acting against us to try and prevent change. Degraded ecosystems are some of the most resistant and resilient ecosystems out there because of the amount of effort that we have to put in to shift their state. And so we think that this could explain a bunch of restoration failure. We've been working on that at various scales. This is just the start of some of this work. Izzy Barrett did another experiment here in large mesocosms. These are about 2 metre in diameter. And importantly, they were flow-through systems, so organisms could leave. And we added EPT colonists, so a desirable fauna that we wanted to get back, to pristine water conditions, indicative of a restored state, a restored abiotic state. But in the degraded community treatments, we had a degraded community established. Much of that was dominated by Potamopergus snails, New Zealand's gift to the world, and in comparison to control treatments which had no community fauna there. And when we looked at the colonisation, the arrival and the density of those organisms after we'd added them in the tanks, the EPT numbers declined much faster in the system which had the degraded fauna present in comparison to the control. And then after 40 days using another sampling method, the number of organisms, the number of EPT left in the tanks and the degraded system was lower than that in the control. This is really indicative of negative resilience, where snails still dominate and the EPT leave when added to clean tanks with a degraded fauna present. Much of this revolves around the snail, New Zealand mud snails, and it likely involves priority effects, facilitation, competition, and predation. They all matter, and they're likely to affect community reassembly under restoration when we fixed the abiotic conditions. I don't really have time to go into all the details of those interactions, but there's a bunch of work done there by Bridget White and Emmy Eggersfield, which documents some of that, published in Freshwater Science as well. Interestingly, this snail, Potamopergus, is becoming incredibly abundant in New Zealand to the extent that it may be, take on native invader kind of status, so it's native to New Zealand, the New Zealand mud snail, but it's becoming particularly abundant and it may be really problematic under these sorts of situations. So this idea that resistance might be a thing in restoration is one which we've tried to investigate more. And Lydia Gilbert, who's an honours student, did a whole bunch of work last year looking at the all-time failure literature that we could find from stream ecology that met a bunch of criteria. She analysed all of those papers and found that biotic interactions are really incorporated into restoration actions, but are often discussed in failure reflections. So let me just run through that a bit now. So if you have a look, she divided this up into four aspects of the papers. First of all, the goals. Many of them mention biotic factors. In fact, there was a criteria, that they had to be addressing some biotic component of the community. Only 13% mention biotic interactions in those goals or the intro section of the papers. Most of the actions, however, 72%, are associated with abiotic conditions. They're only mentioning abiotic factors. Biotic interactions only rate sort of a 5% mention. Monitoring, they all look at biotic factors, but most of them ignore the interactions. But interestingly, when you look at the reflections, and most of these are in a negative context because these are restoration failure examples, 50% of them mention biotic interactions. And so we think there's a real smoking gun there, that we're missing some of the most obvious things to deal with in restoration. Yes, we need to fix the abiotic conditions. Yes, we need to do riparian planting, but we also need to deal with the community assembly processes, all of the community assembly processes which are going on in these kinds of ecosystems. So negative resilience is a thing, it's important, you need to know about it. Thirdly, and going upscale here, multi-scale resilience processes are especially important in complex landscapes like rivers. This is Te Awa Takatamera, the Cass River. It's a tributary of Lake Takapo in the middle of the South Island. And we've been working on that quite a lot recently. It's a big, challenging system. It's hard to work on. It's hard to do experiments in, which I've always loved to do. But Holly Harris was a PhD student. She's a postdoc now. Really embraced that challenge and pointed out how large-scale processes are likely to underpin many of the aspects of resilience in these types of systems and really depend on river heterogeneity. She sampled this river. This is the delta part just before it goes into the lake. Over a couple of years, lots of different samples. If you think about the kinds of consumers which live here, then there are Deleotidium mayflies. This is the most common New Zealand mayfly genus. They're particularly important food for all of the birds and fish which live in this river. But I'm trying to represent here that they're foraging at a small scale. You can see them eating in this forest of algae, and they're moving around at relatively small scales. That's quite different to the fish like this koaro, or Galaxias brevipinnus, one of the native galaxias in New Zealand, which are foraging at a multi-branch scale in this braided river. And those different branches had different stable isotope signatures, so we were able to identify with mixing models where these fish were foraging. And even the ones, if you have a look, the koaro from the lateral springs there are getting more than 50% of their diet from lateral spring habitats, but they're moving into minor braids, major braids, mid-channel springs. That's a long way. They're moving hundreds of meters across this braid plain to get food from different parts of the channel. And the ones in the major channels, which is the bottom set of diagrams there, they're particularly feeding on the mid-channel springs. So they're probably hanging out in the major channels and then moving into the springs. And so that spatial temporal variation in foraging is likely a key stabilizing mechanism in big systems like this, because those different types of channels have asynchronous populations, and that's driven by the heterogeneity of the disturbance going on across the braid plain. There's a bunch of other mechanisms here. This is a picture of a dotterel chick. They have precocious chicks which hatch out and are pretty much able to forage. The parents are maybe a little bit delinquent, or maybe this is, basically they don't do a lot of parental care. The chicks forage themselves. But those dotterel chicks are getting more than 50% of their resources from the aquatic environment, whereas the adults are picking a lot of invertebrates out of the gravel. They might be aquatic adults, but they're picking, they're using different food sources. So there's ontogenetic niche shifts, there are subsidies going on here, and there is omnivory. all of those processes, all of those food web characteristics, if you like, are resilience conferring in a system like this, which is inherently incredibly disturbed. And Holly, through all of that temporal sampling, was able to show that when you look at the variation in the aggregate biomass and you add habitats, this is a thought experiment here, that variance, because of that asynchrony in all of those different habitats, the cumulative variance is much lower when you've got many habitats involved. And so landscape scale portfolio effects likely underpin ecological stability and a loss through landscape simplification. The third dimension that I want to talk about are these landscape, that I want you to remember are these landscape portfolio effects. The last ecological dimension that I want to talk about here is river size. And it's important, it's been important since the river continuum concept and basically since forever water started flowing downhill. And it's likely to influence resilience. We did an experiment here. This was kind of another crazy experiment which involved doing a water blaster disturbance across 10 streams which covered a river size gradient. None of the hire companies in Christchurch like us anymore because we broke quite a lot of water blasters, but because we went to different hire companies, they don't know that, what we were using them for. But we consistently water blasted these streams. Pete McHugh did this. Hamish Gregg was involved in a bunch of the write-up. And we were able to measure various components of stability here. But the decline in the communities that were disturbed with the water blasting compared to control communities is the resistance component. And then rate of change in Bray multi-scaled distances is the resilience component. And both of those are really closely related to the size of the stream, the size of the ecosystem. Resistance is probably most important because the larger rivers were much more resistant. They moved less away from the control, and the smaller rivers moved a lot, and then because they changed a lot, they also had that high resilience component there too. But ecosystem size absolutely affects stability in this context, and is probably underpinned by lots of size-dependent scaling processes. That situation is likely to be really important for restoration. It might be, and many of us probably invoke this, that small streams like this agricultural one on the Canterbury Plains are probably easier to manipulate. They're smaller, they're potentially easier to change. They're also more vulnerable. And so river habitat size likely influences two key things that we're interested in here in resilience, if you like, and we've talked about that in a recent review paper. But that situation really raises another issue that's been talked about a lot at the conference here. I'm talking about, don't forget about river size. And the issue that I want to talk about is the human dimension. And recently, I've been lucky enough to be involved in a research program called Fish Futures. It has a lot of social science involved in it, and it also has 3 rohi partners. So these are 3 Māori iwi groups who've been involved in designing the program. This is on the marae at near Tumuka, the Arawhenua marae. And when you talk about where you might restore and what might be more resilient and might be less resilient, and you suggest that you should focus on those small streams way up there in the headwaters, that's not very popular, and that's not how they see the world. And it's because there's a very important concept which underpins Māori society and views of the world, the Te Ao Māori view, which is called whakapapa. And it's the intergenerational link between people, the land, and the organisms, and the ancestors as they see it. And so if we're to invoke resilience and restoration concepts and talk about that, we need to understand those associations. And Mātua Kao here is on the marae, and he's standing in front of the Wharenui here explaining to the Fish Futures group, he was talking about the name of the marae, and can you see it there? The literal translation is the dishonesty of New Zealand. That's what the name of their Wharenui is, and it reflects the historical grievances which continue today, the loss of traditional fisheries, and the pain and hurt that they've endured. And so if you were to move forward and understand where we might go in resilience, and we heard about this yesterday with the Coeur d'Alene, you have to acknowledge where those people are coming from and where we might go to. So the last thing I want to talk about is this human dimension, and for you to remember. But there's also a wonderful, really cool side of that, and it's to do with these sorts of things. This is, I have to remember this, Macrorothalmia stage of Kanakana, a New Zealand pouch lamprey. I worked in freshwaters in New Zealand for 30 more years. Until a couple of years ago, I'd never seen these. This is the migratory stage, which goes out to the sea. I have this wonderful iridescent blue colour. I only got to see these and understand about them because Jane Kitson, a wonderful Māori scientist in Murihuku, explained where they live, enabled us to figure out where we could sample these. I've got interested in that. We've started this thing called Operation Kanakana, which is about the restoration of Kanakana populations. It's a really important fishery. Started to search out the ammosetes, try and figure out where they live. They live in the gravel. They look cool. They're really neat. And so there's this other side, which is really amazing, that co-development enhances resilience planning. It's great, it's rewarding, and it's essential. And so I really encourage you to embrace those dimensions, and it involves working together in just ways. I wanted to finish up with this. This is a puhoro. The design is shaped to represent the flow and current of all water. It's also the pattern left in the water of people paddling a waka, a canoe. You can see that on both sides there. But also in the form of a wave, it represents movement, innovation, and vitality. And we can also see a traditional pattern of fish scales and waters in the design, and that it also represents the well-being of people and water. This is the new symbol of Ngā Kahingawai o Aotearoa. I'm incredibly proud to kind of embrace what this represents. It's a real privilege and an honour. It needs to be respected, but it's also amazing to be part of that. And to me, it represents much of what I've been trying to suggest today, that working together, coming up with innovative ideas. The world's a pretty screwed up place at the moment, and if we can work together and work for good, then there's a lot that we can do. So with that, I want to thank you for listening. Thanks very much for inviting me here. It's wonderful to be here. Ka nui te mihi kia koutou. Kia ora.
And I'm now honored to introduce Faith Kearns, our second speaker this morning. She is the Director of Research Communication for the Arizona Water Innovation Initiative at Arizona State University. She earned her PhD in Environmental Science, Policy and Management from University of California, Berkeley in 2003, where she worked with longtime SFS member Dr. Vince Resch. She also told me that she presented her first professional poster at a NABS meeting in 2001, so long history. She served a term as a AAAS Science Policy Fellow with the U.S. Department of State in 2004, where she negotiated international ocean and fishery policy. Her career is focused on science communication and engagement with an impressive publication record that intermingles publications in peer-reviewed journals with media such as the LA Times, Newsweek, National Geographic, Washington Post, and on and on. Her popular writing addresses water and drought, wildfire, and climate change. She's worked as a professional science communicator for diverse organizations, including the California Water Resources Institute, Pew Charitable Trusts, UC Berkeley Fire Research and Outreach Center, among others. She co-created and co-hosts an amazing podcast called Water Talk that has 6 very meaty in terms of content and number of episodes seasons. And in 2021, she published Getting to the Heart of Science Communication, a guide to effective engagement by Island Press.
Got some notes up here. All right, thank you so much, Camille, for the introduction. It's really good to be here with you all. Thank you for getting up early and coming to listen to these talks. So I think Camille did a pretty good job introducing me. The only other thing I would say is that I spent a lot of my career working in California's Cooperative Extension Program, and so you'll definitely see the impact of working in cooperative extension in what I'm about to talk about. So let's see here. So I'm going to start out with a little bit of a story, just to give some context for what I'm going to talk about. So about 20 years ago, getting older, I was not that long out of graduate school when I went to work in a fire research and outreach center at Berkeley. And I had this pretty sort of career and ultimately life-changing interaction with this man at this workshop, because we were, in this small Northern California town doing a really pretty common thing, which is to set things on fire and show people how their houses burn down and things like that. But part of it was also that we were, there were a few of us there and talking a little bit about some more challenging issues. So, we were doing pretty cutting edge research at the time. It's seemed very controversial for some reason, but now is pretty much what people would recognize as the standard of, how we think about fire-resistant building. And so we were sort of doing all the things we were supposed to do. We were developing this research. We were making it available online. We were in conversation with state legislature, local fire departments, the state fire chief, et cetera. And so, we sort of thought we were doing everything we needed to. But that day, we also talked about a very controversial idea of a stay or go policy that we were ultimately writing a peer-reviewed paper about, which is the one you see there. And the idea is that when there is a wildfire, people can be trained to stay with their homes while they have a fire raging around them, right? This kind of practice is fairly common in Australia. They have a very different firefighting system than we do. And it's just interesting because at this point, that is becoming a very sort of full circle issue. The actual CAL FIRE is actually talking about training people to do this 20 years after we wrote this paper. So, but when you're listening to this and imagining it, I think it can bring up a lot of very sort of vivid feelings, like literal life and death feelings. And in a story that will sound pretty familiar at this point, but wasn't at the time, we were in a place where if you look, let's see, Big Sur down on the sort of central coast and then up in Mendocino County. We were up in Mendocino County, and it was in the fall, and earlier that summer, this situation had happened where there was a whole big set of fires in Big Sur and then Mendocino, and the people in Mendocino felt like they were not getting attention from the fire department, and ultimately a lot of people had to be evacuated, and there were like 18 other fires all over the rest of the state, which we can see happening, starting to happen just today. So all to say that emotions were running really high in that room. People were sad, they were angry, they were anxious, they were sometimes grateful that the damage to their property hadn't been worse. But this man that I was talking to, basically, in words that I would use today but didn't at the time, was essentially telling me that our presentation had brought up essentially trauma and PTSD, because we were literally talking to people who had just sort of experienced the very thing that we were talking about. How do you decide when to evacuate or not, you know? And the crazy thing is that none of us going into that day had at all considered that this community had been on fire a few months before we were there, right? And so really, I'm not sure how that interaction happened, but it was, you know, I ended up being very sort of present to what he was telling me, and that one conversation basically shifted the entire way that I think about science communication and community engagement. And I think it's just that experience of sitting and listening with somebody and realizing that the research you do actually does have sort of real world implications, right? You know, things have only gotten worse in this regard. I did some early work on wildfire and its impacts to water supply and infrastructure. Wrote a report in 2021 or so, sent it around to some journalists. Nobody was that interested. And then, of course, came the LA 2025 fires, and suddenly we have been able to propel a lot of that work forward, but it's still a really challenging time. I don't know if people saw how much mis and sort of disinformation was going on about water in particular during the LA fires, and so it's been a really interesting and controversial topic. So to get into resilience, right? So this kind of sums up my perspective on resilience, right? And the meme is funny because it's true, right? So I'm not going to get into a long old definition about resilience or anything, but I think the idea and the way that it has been presented to people has been very much around like learning to withstand harder and harder conditions, right? You're so resilient, making it through your very difficult conditions, right? But what I'm really going to talk about is sort of resilience that comes from relationship, connection, you know, structures for making that happen. So I'm going to talk a little bit about science communication just because I find a lot of the sort of disagreements about science communication are largely because we're working from sort of very different definitions of what good science communication is. And so for decades, all of my career, basically, the dominant model for science communication and training in the United States in particular was very much focused on the idea of performing, right? Kind of what I'm doing right now. The expert on the stage, somebody filling this information gap, and the assumption of that sort of what we call the deficit model was that if people just had enough information, they would do the thing, right? That's obviously not true. And that model really put a lot of pressure on scientists to be authoritative, certain, unemotional, and again, it hasn't worked out particularly well for us. And so another big thing is just that the communications landscape itself has changed vastly, just even in the last year, which really complicates kind of doing this kind of work. It puts you in a pretty challenging position. So, you know, that same shift is kind of happening in all of these realms that I'm talking about, right? Science has sort of moved on from the lone genius, not to say that it doesn't happen, but a lot of the work that I see these days and participate in are very collaborative, very interdisciplinary, transdisciplinary, community-embedded work. All of these photos are from community engagement events that I have been a part of. And our understanding of resilience is also kind of making that same shift from this very individual focused, you know, being resilient to like what it means to do that in community. And so, you know, these aren't separate trends. They're all kind of moving along. And I also just want to acknowledge, you know, for the early career folks in the room, this is an extremely challenging time. Like, it's extremely challenging for me, and I think if I were sooner in my career. So I just want to acknowledge that. And so my ultimate thesis, especially for my book, was just very much that we face less a science and science communication challenge than a relationship challenge. And my sense of that is that we really have honed in on this idea of how to communicate better. And the last year or two has also brought this up again, like all our problems are because we're not communicating properly, right? And I don't think that's true. I actually think that the communication challenges are sort of a microcosm of what's happening at a larger level. And really, relationship is the container for communication, right? And we just have not invested in that in the way that we probably should. And so what that shift really means is that, that we need new skills. And so I'm going to talk just briefly about sort of sources of resilience for scientists and science communicators. So I think one of the key ones is relationship. I've been having some pretty profound experiences doing community-engaged groundwater work in Arizona, where people, you just see the system that people are going through, where they are realizing that nobody is really coming to save you. The federal government isn't there, the state government isn't there, and then there's this interesting shift where people get kind of depressed, and then all of a sudden they feel very empowered to do what they need to do together, right? So. I think, Theopia Jackson, she's a clinical psychologist and definitely one of my heroes. When I interviewed her, she said, I think of my practice as a service model where I frame my efforts around being in service of others, and that means I can't come at it from a place of being an expert with all the answers, right? And that is a really challenging thing to the way that we teach and train people with science communication, because we're often training them to be the expert with all the answers. And that works in certain settings, but it doesn't work all the time, right? And so the idea is that we shift from sort of explaining or correcting and into connecting. Like there's these phrases that are starting to become sort of part of the way that people are talking about things these days. So just to give a specific example of what that looks like. So here are two people in very, very different conditions. Sarah Watson, when I spoke with her, she was working for Sea Grant in South Carolina. She's now working for the state of Connecticut, but she told me, I give a lot of presentations on the local effects of sea level rise to groups like homeowners associations. Each time, at least half the people there are completely freaked out. I now get nervous having to say out loud what these climate projections tell us about flooding in the region. It's hard information for communities to hear for the first time, and it's hard for me to give the same bad news over and over again. News I increasingly worry isn't bad enough to cover where we're headed, but simultaneously, worry sounds too alarmist for what people are ready to hear. And Mila Marshall is a water expert in Illinois who told me sort of a similar thing, which is like when you're talking about difficult subjects, we were primarily talking about vaccine hesitancy at the time because it was early COVID years. You have to understand you're asking people to acknowledge there's a chance something they believe to be true, told to them by people they love and respect, is wrong. We can provide accurate information, but there also has to be someone standing in the gap, prepared to support them, right? And I actually still think this is such a brilliant idea to actually have people who don't just deliver information, but also can help people process that information. That's essentially what happened with me in that fire workshop. Another practice that I am a big fan of is, listening. And there's lots and lots of ways to listen. It's becoming something that people are talking about more and more in the sciences. And I think I have been able to luckily learn a lot from folks like Jana Lambrinadu, who's an anthropologist at Smith, who's done a ton of work on listening and is the only person I know who's taught a listening class to scientists and engineers, a full class about how to listen. And so what she told me was, listening is often diluted to become about empathy and compassion when it's about justice and accountability. How scientists listen is inseparable from questions of power because it can be done in ways that bolster community knowledge, but it can also be done in ways that compromise scientists' ability to help in desirable and just ways. And a big part of why Jana is talking about this is because she worked a lot on the Flint water crisis. Something that generally makes people uncomfortable, especially the science types, is conflict. And so we tend to really, lean a little hard into conflict, right? Like people just don't want it, they just don't want any conflict. And so the thing I really am hoping to do is just normalize that doing good work involves conflict. Collaboration is not the answer to conflict, that conflict still comes up in collaboration, right? So for me, the goal is just for people to understand conflict as a normal part of human relationship. It's, it's always present in the kind of work we do. And I think the biggest thing that people can do, myself included, is really just get more comfortable sitting through conflict because there's often a ton of information in conflict, and if we're just fleeing it constantly, we are missing a lot of things. And then the last sort of tool in the toolkit, I think, is just understanding trauma, being trauma aware. Obviously, I work on a ton of disaster issues, and this is something that's extremely present for me, but I think it's extremely present for everybody almost at this point that I talk to. Miriam Kia-Keating's a psychologist at UC Santa Barbara, and one of the only people I know also who has really studied fire survivors using ethnographic methods and has found some really interesting stuff. But she said, just understanding there is a psychological impact from disasters is valuable. Recent events have opened my eyes to the personal distress researchers and professionals have been experiencing. Those who help communities respond to disasters can experience secondary or vicarious trauma. So this is work that is extremely challenging, and we're also living what we study, right? I myself have had to evacuate a fire or two, and this is a quote from Tessa Hill, who's faculty at UC Davis. This is a quote from after the Tubbs fire in 2017, and she says, we awoke to multiple friends asking us if we were evacuating. We launched ourselves out of bed, turned on the TV, pulled up social media feeds, and began a frantic hour of messaging friends and family, trying to understand the wildfire chaos around us. By daylight, we noticed some of the ash was as big as our hands and had legible writing on it. Other pieces falling into our yard were burned fabric. The remnants of our community were falling from the sky. That fire erased our sense of safety, not just at the individual level, but across our county. It was a community scale trauma, right? And so how we navigate the things that we are also experiencing, I think it makes it even harder to think we somehow can be objective in these situations. And I think the response to all of this really is solidarity, right? Like the further and the further I go in my career, it's just like rules, certain types of infrastructure, whatever, they're all there, fine. But at the end of the day, you really need other people. And so, I think it's something that we have to work at a little bit, and honestly, navigating solidarity is really challenging too, because in a certain way, if you align yourself with a person or an issue or whatever, that means you're not doing it in this other way, right? And so it's this constant negotiation of what we can do to benefit everyone. And to not leave this on like a totally downer note, I do want to talk about, science communication certainly can be joyful. It's challenging in some of the work that I do, but I see other people having more fun with it. And so my colleague Malika Noko, who also co-hosts Water Talk with me, was talking about a fellowship retreat that she went on, and they had to look through all these public comments, and they were trying to do a research project on conservation, and she said it was so depressing that at the end of it, like the Friday night when they were ending this thing, they just decided, hey, let's create this thing called Plant Love Stories, because we wanted to focus more on plant appreciation than science and just make it fun. It makes me feel joyful and light. It makes me feel like I can be funny. I have all these parts of my personality I feel like I have to suppress for science, but with this I can be goofy and make puns and be a total plant nerd, and it's okay. So really, I guess where I want to end is just to say that, one thing I especially think early career folks can take on to not get burnt out is that there was a very well-known book that talked about the idea of good enough parenting in like the 60s and 70s, right, which was sort of this antidote to other more stringent kinds of things about raising kids. And the author talked about this idea that there's just good enough parenting, right? And I think that concept also applies in many other realms. Like we cannot be in search of perfection all the time. So some concrete practices, resilience isn't abstract. It really is sort of built through practice. It might not even be the right framework, but I still think it's worth thinking about in terms of just how we can stay in this work for the long term, right? So sometimes when I give these talks, it can come off that I don't think that knowing your shit is important. I would say that's absolutely not true. I think it's very difficult to navigate these kinds of issues without being very confident in your or sort of baseline knowledge of the stuff you're working on. So, school's still important, et cetera. But there are certain things that you can sort of do, practice like putting distance between yourself and your reaction to things, not suppressing, your emotions, but just learning how and when to express them. And I think, seeking out any kind of community of practice these days is so important, more than it ever has been. And a kind of random note, in this day and age, I think, we're a society that's very much moving away from reading, particularly reading books. But I think it's such an important thing in terms of just cultivating imagination and compassion. And I know when you're, especially in graduate school, there's this sense that you should never read anything other than what you're supposed to be reading. But I think, again, like if the goal is to sort of like train people broadly, the inspiration for my book came from reading so much medical literature, stuff about, the way psychotherapy works, all sorts of stuff. And like I said, give yourself permission to be imperfect and keep going, because we really do need everyone in this work for the long haul. And so I'll just end on this note that, staying human is more important than ever, I think, and is not a liability in science or science communication, right? It's sort of the point. So these, resilient freshwater systems that Angus was talking about, and the resilience of the people who study them, everyone in this room, go hand in hand. So, I'm just going to end there. Thank you. Thank you for listening. It looks like we've got about 15 minutes or so for questions, so if people have them for me or Angus. Should we take this out?
Hi, Dr. Kearns, that was a fantastic talk. I loved all of it, and I have so many questions. But I think the one that I want to ask has to do with capacity in light of the incentive structures, both in terms of in an academic career, which is where I'm coming from, and then in response to like funded research projects, right? Like those kinds of relationships and one-to-one communications, valuing presence over performance, is clearly really impactful and really important and really meaningful, but really difficult to quantify and demonstrate, and thus difficult to incentivize when you're trying to decide how to spend your limited time as an early career researcher, trying to get tenure or get funding or whatever, and trying to convince your funders to fund you again. Whereas those performative things, like look at this, cartoon I made that posted and 10,000 people looked at it.
Yeah.
So I don't know if you have any advice for how to kind of balance that or
Yeah.
make that work visible, because it is true work. It takes a lot of time.
I think it's super challenging, right, for the academics in the room. I think, you see all the time some effort at reforming the incentive structure, and people are more and less successful at it, and institutions are more and less accepting of it. I see lots of calls to recognize public scholarship in terms of people's incentives. But what I would say as a practicing science communicator is actually just hire people to do science communication. If we care about this stuff as much as we say we do, every time I open there's always some cry. We're just so bad at communicating. Well, we don't really invest in it. We expect you guys to do it, with the 5% of your time and then not get rewarded for it. The best communications and centers and all these things I've seen on campuses, they do well because whoever is doing them is incentivized to do that thing. And it may never work out for faculty to be that. The further I go in my career, I don't see it happening. But I do see a class of professional science communicators rising up and really just need jobs. There are lots of people who can do this work. It doesn't always have to fall on faculty. Yeah.
I was nodding rapidly. You probably didn't see me, but I was going to say that in building relationships, that takes time. It doesn't happen. It doesn't. It's we need to kind of shift the focus of academia, towards outcomes instead of outputs. I would add knowledge brokers to the communicators part. Our grant proposals need to add that, and our university structures need to shift to, reward those things more. And that's something that I take pretty seriously, putting my associate dean hat on. And the more that we can, bring about that change, the better. But it needs to happen soon.
Yeah, those were really interesting presentations. I wonder if you could both comment about the role of community science nonprofits in building resilience.
Yeah, absolutely essential, because I think we can do science, and quite often the scale of that science is very local, it's limited, but being able to spread that and make that be useful at a really large scale very often depends on those non-profits. And many of the iwi groups in Aotearoa, New Zealand, are run on a shoestring. The one who's associated with the Kanakana work down the south is Oraka Aparema. They've got a very small number of kaumātua. The resource is small, but the work that they do, because they're connected with farmers and they have relationships in the community, if we can be in relationships and be involved in those, then they have, through that association, there's the capacity for our knowledge to be much more useful.
Yeah, I agree. I think a lot of the work I do is working with other groups outside of the university, and it's hard for me to even picture not working in that way anymore. And so I think there's a real synergy with what a lot of the NGOs and community groups, community science groups are doing. For example, in Arizona, as part of our groundwater work, we just went through a big establishment of these things called libraries, where you can go check out equipment and look at your groundwater. And so super supportive of those kinds of relationships, for sure.
I was going to say in response to that there's also, that's a pretty big source of conflict, actually, Faith, that as a scientist I've quite often gone into a room with a group of stakeholders who have a particular idea about how they want to restore a stream, and I quite often have different ideas, and how you negotiate that is absolutely the crux of what you're talking about. I think understanding that the relationships and trust and building those comes before being able to negotiate, find a solution to those conflicts is important, and that's really important with all of those stakeholder groups, I think.
Hello, I have a question for both of you. I'm back here, sorry. I would love to know how you have or haven't altered your communication strategies to incorporate all of the new digital spaces and digital platforms over time, because there's always a new one. So I'm just curious about that approach.
Yeah, I mean, I've been doing science communication since before it was even called science communication, so I've seen a lot of change about how we do these things. And I will say the last year or so has been the most challenging kind of space we could be operating in. And so in terms of, I just was reading somebody the other day who's sort of we're not in a 2.0, right? The internet, social media, et cetera, it's just integrated into everything we do. So I don't really consider it that separate from the rest of the work. But I've done tons of web materials, social media stuff over the course of my career. And I think where we are right now, I would actually like to get sort of a working group on it, because as everybody probably knows, these AI summaries at the top of your search are ruining science communicators' lives, because you're often trying to point people to information, and now they can't even find the source of that information because these AI summaries come up and people just kind of glance through them. They don't necessarily click the links. So everybody who is a sort of information provider in the digital space is seeing dramatic drops in web traffic, and it's changing the way we think about incentives. I might still be really good at my job, but like nobody knows how to crack what's happening right now in digital spaces. The other thing that I think has been so prominent, and I'm still trying to wrap my mind around, is how many people are participating in these sort of smaller communities, like Discord servers and whatever. And so the big sort of public conversation that used to happen on Twitter has really sort of devolved into these little niches, and you just aren't able to have like a coherent conversation across these groups most of the time. And I'm not sure what the answer is. We're really right at the beginning of trying to figure this out. So,
yeah, I'm not sure that I'm the person to answer this question, because there was last week I was in a tutorial, and I suggested to the students they should make a tutorial group on Facebook or something like that, and one of my colleagues said, well, students don't use Facebook anymore. I was like, oh, okay. I think, yeah, absolutely endorse what you said, Faith. And I mean, we've got such important messages about how there are interesting, cool, exciting things that live underwater. And to me, through photography, through teaching, through science, one of the ways to be effective in that is to try and bring those stories, out. And there are conflicts and things that we can do, but if we have a shared basis for conversation, I think that can work. The question is, what's the platform? How do you do that? I think we're all struggling to figure out how to do that. The thing that I'm consistently told, particularly from iwi groups, Ara Whenua in particular that we're working with, is that they need something that's accessible. So one of the consistently most difficult pieces of writing that students do is for those sorts of groups. It's really hard when they have the consistent science training to write in a particular way to switch to a different way. And I think there needs to be a fundamental switch, where that is incorporated in right from the start, that we're writing for a number of different audiences, and many of those audiences are not the general public, because I don't think the general public exists. It's who is the audience, and can you write for this particular audience, which is, this non-scientific group.
Hi, thank you both for your talks. It's not often we get to see such information juxtaposed in terms of biological understanding of resilience and a deep understanding of human relationships and resilience. And so if you two are both practicing active listening to your talks, I'm going to ask you to do some work. Angus, what would be something you learned from Faith that was about how human resilience is built that you could now apply to a biological understanding, and vice versa?
Great question. Actually, interestingly enough, I, in my Dean role, am about to give a talk in 3 weeks to the New Zealand Science Communicators Association that's on campus of my university. So I was like furiously writing notes, and they're all down there. So you really put me on the spot, Kirsty. But I really took out the relationship part, and you've heard us, talking about that, and it absolutely just works true. It works true in mentoring. It works true in teaching. It works true in working with community groups. It works true in with iwi groups. Things revolve around relationships. I also really appreciated the idea that this is difficult, that there's trauma, and I like the idea of embracing conflict as well. I think that's for those mentors and students out here, there's quite often conflict in those relationships too, right? But being able to negotiate that and work through it in a way that ends up being positive is really important, because there's lots of different points of view out there, and if we can understand and negotiate those, then that's important. I mean, what's really challenging, in the world that we live in is the restructuring of science. We've just had a whole bunch of funding reduced, moved, reallocated, taken away last Thursday when I was leaving New Zealand, trying to negotiate that and help faculty, understand how to do that. There's trauma, and it's difficult.
Yeah, I would say I would say your presentation gave me a chance to just kind of go backwards for what I used to, how I started out studying things, which was very much counting stream bugs, et cetera. And to think about where the overlap with sort of our scientific definitions of what resilience is, and then how people are talking about it more broadly, is sort of interesting, because I don't know if there's quite the same value judgment around resilience when it comes to ecosystems as much as there is with humans. Like, we must all be resilient to whatever is coming our way. And so it's just, yeah, it's been interesting to re-immerse a little bit and think about, you know, the parallels and the non-parallels, I guess, yeah.
Thank you both for your talks this morning. My question is about the tension between trauma to human communities versus the importance of disturbance regimes for ecosystems. In the US, there's a history of extreme wildfire, extreme floods that was responded to with suppression, fire suppression, and flood control that in some ways we're paying the cost of that now. And I'm wondering if you're seeing that tension play out and how that plays a role in the conversations you have today.
Yeah, absolutely. That's huge in the Canterbury Plains, the South Island of New Zealand, where we have these really big braided rivers. They formed the plains. They moved across. They're now been turned into agriculture. They're full of irrigators. We have put all of these flood protection works, and that's starting to produce a lot of conflict because the constraints of the channels reduce the heterogeneity, likely removing, simplifying those channels, undermining the natural resilience that they have. But at the same time, as the flood magnitude gets higher, places the communities behind them at much higher risk, because they're likely to overtop more. And so there's this difficult negotiation around the idea of giving rivers room to move, which is really important, about how we negotiate that. How do we move the stock banks back? Do we buy back land? In many cases, some of that land's being taken illegally, that the farms that just moved out onto the Braid Plain and parts that weren't flooded, in the last 10 years have been turned into farmland. And so how all of that gets negotiated and sorted out is challenging, right? But it's for river ecosystems that the capacity to move and, that absorbing capacity that Erin in that conceptual paper talked about from a geomorphological point of view is really important, I think.